Hello everyone and welcome to our today's webinar, Steer Tank Bioreactors and Vaccine Development, Producing AAV Particles and COVID-19 Spike Proteins in Suspension Culture. My name is David Solbach. I am the Global Marketing Manager for our bioprocess products at Eppendorf SE, and I will be your moderator for today's webinar. If you want to know more about our Eppendorf Bioprocess Solutions, visit our website at www.eppendorf.com/bioprocess. I encourage you all to participate today by submitting any questions you might have during the presentation. But before I will hand over now to our great speaker, Jorge, today, I would like to do my job first, actually, and ask you two short marketing-related questions. So everyone who is joining now, you should see the first poll question, and I would like to know from you, before you registered for this webinar, just curious to know if you were aware of our Eppendorf Bioprocess Solutions. I will give you some time to realize that it's not listening only, but also some work for you to do. But no worries, it's just one out of two questions, and the only other thing you need to do afterwards is listening to the presentation and type in your questions. And while we are waiting, I can try to view the results. So, thank you for the first answer. Let me quickly check here. For you, interesting as well to see maybe, so it's nice to see that at least the ones who realize that you're not only listening today, but also working, answered the question with a yes. This is, of course, really nice to know. The second question for us is, I would like to get to know you better in terms of if this webinar you registered for is also helping you. So which of the following answers best fits to your current situation? Are you working or doing research and development currently in 2D systems, or are you already working in 3D systems, or are you considering to switch from 2D to 3D? So take your time, have a look, and feel free to answer. If you are doing research and development in 2D systems, I'm pretty sure you will learn a lot today about how steer tank systems can improve your process. If you are already working in steered systems, listen to Jorge and take the opportunity to ask any question you want to know to improve your process and to get expert hints. And if you are considering to switch from 2D to 3D, the same is true. Jorge will answer you all your questions. Here are the results so far. Some results are still popping in. I'm not sure if this is updating after it's pushed live. Thank you for your answers. And with this, I'm already done with the, let's say, marketing part, and the scientific part will start now. I will hand over to my colleague Jorge. He is our cell culture as well, and cell and gene therapy expert in our applications team in Enfield, and he did most of the work, or all of the work he will present to you now. So Jorge, the stage is yours.
Thank you, David. My name is Jorge Escobar, and it's certainly a pleasure to be here today and welcome everyone to Steer Tank Bioreactors in Vaccine Development, producing AAV particles and COVID-19 spike protein in suspension cell culture webinar. So today I'm very happy to introduce some of our newest applications in vaccine development. So I'm going to start with some introductory slides about vaccines, the state of the art, benefits, and industry growth. Also, in this talk, I will give an overview of some of the most important vaccine platforms. And finally, I'm going to introduce most of the relevant information about two vaccine applications that were developed recently in our application lab. One related to adeno-associated virus, AAV vector production, and the second one about the SARS-CoV-2 spike protein production. Let's start. So as many of you know, a vaccine is a preparation used to stimulate the body immune response against diseases. So it can be administered mainly intramuscular injections, but oral and nasal administration is also possible. So vaccines help eradicate diseases in the past and today prevent between 2 and 3 million deaths a year worldwide. As you see in the slide, data from the CDC shows that how effective vaccines have been in eradicating major diseases in the United States. For example, in 2020, there were no reported cases of smallpox or paralytic polio, for example, compared to the annual average of 29,000 cases and 16,000 cases in the 20th century, respectively. So, a wide progress in eradicating measles as well has stalled in recent years, probably due to the growing skepticism about vaccines, and its morbidity is nowhere near to the annual cases seen in the 20th century, when 500,000 people were infected in the average year. So it is well known that pandemic outbreaks have occurred intermittently since times immemorial, damaging communities socially and economically and causing large number of deaths. The image represents the timeline of various contagious viral outbreaks in the world, from the Spanish flu to the recent COVID-19 pandemic that has been infected nearly 620 million people and killed more than 6.5 million people so far. So the new coronavirus is less deadly, but so far more transmissible than MERS and SARS-CoV. And for sure, the COVID-19 vaccines have been effective preventing and protecting people from getting seriously ill or being hospitalized, and also protecting people against new variants. According to this analysis, market analysis, the vaccine market size is expected to be valued in $28 billion by 2026, with a progressing growth of 4.13%. So the largest segment of the market are prophylactic vaccines and therapeutic vaccines. And nearly 48% of the market growth will be originated in North America, specifically in the US and Canada. But you will see that this region, the growth will be slower than the growth in the market in the European and Asian region. And companies like Stella Pharma, AstraZeneca, Merck, and others will be or are the most important players in the field. Now I'm going to give an overview of some of the most important vaccine platforms. So this schematic representation shows different vaccine platforms for infectious diseases, showing different vaccine technology against viral pathogens. Vaccines can be classified as live or dead vaccines, and also according to the platform used in their manufacturing. So as you can see many examples in this picture. Conventional vaccines use antigen derived from disabled weakened pathogens or their components, such as subunit protein, to generate an immune response to possible future infections. Specifically, the protein subunit vaccine, instead of using the entire pathogen, they include components or antigens that the best stimulate the immune system. So this strategy often require the incorporation of adjuvants to elicit a strong immune response, because antigen alone are not enough to induce long-term immunity. Also, eventually, the antigen is digested, but antigen present in cells, such as dendritic cells, as you see in this picture, which present the antigen for recognition by the immune system. Then the T cells, the T helper cells, will recruit another immune cell, such as the B cells, which will produce specific antibodies against the antigen. Then these antibodies will attack, the virus that is, in the host cells, and they will attack the pathogen for destruction by macrophages also and killer cells. On the other hand, we will have also B and T immune cells, remain in the body after infection to provide immunological memory. As you see, many FDA-approved vaccines and other candidates in preclinical and clinical trials in the world are produced using conventional vaccine technology. So this is the case that, for example, the live attenuated, the whole inactivated and recombinant subunits, and also some candidate recent COVID-19 candidates that have been produced using this technology. However, this strategy is often not sufficient against complex pathogens like this our recent COVID-19, and they can evade the immune system and also provide short-term immunity that require repeated inoculation and take more time to manufacture. The development of vaccines against emerging pathogens with pandemic potential is not feasible using this conventional technology, as I mentioned before, and also due to the manufacturing problems. I think that we have learned some lessons from the recent COVID-19 pandemic. For example, that the global manufacturing capacity was limited to answer to somehow answer the demand that the patients needed. And also, consequently, we must be prepared for future, but the unknown remains the greatest challenge. So we don't know what is coming. As a result of that, it is really important to develop platforms that can be applied rapidly to respond to pandemic threat, and that are also effective against emerging variants. Another important to highlight is the scale-up capabilities. They should be ready after vaccine development, selecting the most efficient platform to increase the vaccine production in most geographical areas. Viral vectors for gene therapy and the use of nucleic acids like messenger RNA vaccines have enabled to develop novel vaccine platform to meet the criteria against emerging pathogens. The aim of both technologies is to transfer fragments of this genetic code taken from the target pathogen into the patient cells, and the cells will produce the antibodies and induce an immune response in the body. So one of the next generation of vaccine platform is the viral vector vaccine, and is one of the most popular platform in gene therapy today. The viral vector vaccines are viral particles unable to produce or cause any disease, and they don't pose any hazard to the human body. These viral particles contain the gene of interest, as you can see in the slide, of the select pathogen, and this particular genetic material is transferred into the cell by the vector to induce the immune response against the chosen pathogen. So I would like to highlight this, that these viral particles are called capsids. But how does the viral vaccines and the viral vector vaccines work? So as you know, all the vaccines are designed to introduce antigen to the body. So some vaccines train the cells to produce antigen themselves. And this is the case of this viral vector strategy. The vector is delivered in the body, and the capsids fuse in the cell membrane, releasing the genetic information. Then later on, the cells will read the instruction and express the target protein that finally is exposed in the cell surface. The immune helper cells trigger an immune response and recruit B cells to produce antibodies against the target protein. And and finally, this antibody tag cells display in the protein and destroy them. And also, there are long-living memory cells enabled to switch any response for future infections. Also, another example is the RNA vaccine. It's another really example of the next generation of vaccine platform. I think that we all know a little bit about the mRNA vaccine and how it works, but let me talk a little bit more about it. The messenger RNA is the cell blueprint for building protein. It's a single-strand RNA molecule that encodes genetic information from DNA to be translated into functional protein molecule. So the combination of this RNA with the next generation of drug delivery system, such as lipid nanoparticles, really facilitate the delivery of the mRNA in the cells. And also, this is a huge opportunity and a great platform for the delivery of a wide range of next generation of drugs to patients. But how does the mRNA vaccine work? So it's pretty similar to the vector, but as I mentioned, the mRNA will be delivered to the cells using the lipid particle. And then also the entire mechanism is almost the same. So again, the idea is to train the cell to produce antigens themselves. Again, the immunohelper cells trigger an immune response and recruit B cells to produce antibodies against the target protein. And finally, the B cells produce antibody and they tag the cell displaying protein and destroy them. So there are some examples of viral vector vaccines and RNA vaccines that are already approved by the FDA, and some of them are very popular today. For example, the Moderna COVID-19 vaccine, the BioNTech Pfizer COVID-19 vaccine. We have the Johnson & Johnson COVID-19 vaccine that was developed using the viral vector strategy, and then we have the Ebola vaccine. Well, developing preventive solutions through new vaccine platforms that responds to new diseases is really an important challenge today. In order to produce high-quality biotherapeutics, process development involves certain demanding components, as you see here, including cost, cell line development, small-scale exploration, effective scaling, and optimization of upstream, downstream processes. Vaccine production can be accomplished with either adherent or suspension cell line, but each option really requires a specific design in upstream and downstream processing. When you cultivate using anchorage-dependent cells, the procedure is really more labor-intensive, and the attachment culture lack of large-scale bioreactor manufacturing solution if you compare that with suspension culture. Also, the cell dissociation step is in the process is, will complicate the scale-up process and imply higher costs. We really believe that in order to meet the growing demand of vaccine, adapting encourage-dependent cells into suspension cell culture is a manufacturing strategy that could really simplify the upstream workflow and increase tighter yield while reducing costs. The process development involved in the process, it's important to the selection of the host cells, and it's one of the key factors. And it's important to know its capabilities and properties. So including its ability to grow even in suspension or adhere to a substrate. So as many of you know, a human embryonic kidney, HEG, as everybody knows, for example, have a wide range of advantages, including low maintenance, rapid proliferation, robustness, and convenient application to both transient and stable expression. The HEG cell line, the HEG293 cell line, is one of the most versatile mammalian cell line with a wide range of application, including expression of recombinant proteins, antibodies, and viral vectors. So more scalable vaccine production strategies, of course, are needed in order to meet the global demand, as I said before, and especially on the current COVID-19 pandemic, and with the increased risk of exposure of new pathogen. To this end, we uses a suspension adapt A293 cell line that is called XP293F cell line as a cell culture platform in the development of two examples of bioreactor-based vaccines application. So one of them is the adeno-associated virus vector, and the other one is the SARS-CoV-2 hexapro spike protein production. So in the second half of this presentation, I'm going to introduce details about how we establish a suspension adapt A293 cell line as a cell platform for vector and spike protein production using our newest controller, Siberia Twin bioreactor control system. So the application note 447 from 2021 demonstrates that a suspension adapt A293 is a robust suspension cell line capable of achieving high cell productivity. And in this case, we were able to get 13 million cells per milliliter in 9 days. I really encourage you to and invite you to use this QR code and download the application notes. So you will find a lot of information and really details about this development. So let's start with the AAV vector production platform example. The content of this example is collected also in the application note 450 of this year, entitled Adeno-associated virus production in suspension cell culture using Siberia Twin bioreactor bioprocess controller. Again, using the QR code, you can download information, and that will be really useful. You will find really detailed information about what I'm planning to explain now. So we have previously described how vector-based vaccines work. Let me explain in detail the AAV capsid production strategy. So this is as you see in the slide, this is the capsid production workflow. The process includes several steps like plasmid selection, cell expansion, plasmid transfection, viral vector production, isolation, and quantification. So in this case, we decided to produce AAV2 capsids because are the most widely used vectors in preclinical and clinical studies. Also, this vector has high safety rating due to the fact that the wild type AAV is not currently known to cause any disease in vivo. So as I mentioned, the first step is the plasmid selection. The AAV vectors production involve three plasmids: the transfer plasmid, the Revan cap plasmid, and the helper plasmid. So in the case of the transfer plasmid, this plasmid contains basically the gene of interest and is used to amplify or produce many copies of this gene during the transfection process. The Revan cap plasmid is the plasmid that contains the AAV structural and packaging genes, the replication, and the capsid genes. And finally, the helper plasmid contains the adenovirus genome that is necessary for recombinant AAV packaging, regardless to the Revan cap genes. Two important details here that I would like to highlight. We have used in this application only two plasmids, the Revan cap and the helper, because we wanted to produce empty capsids. And also we use the adeno-associated virus helper-free system. And again, you will find more information in the application note. But essentially, the helper system eliminates the requirement for wild-type adenovirus co-infection and only provides some adenovirus helper factors for AAV packaging. The second step was the cell expansion. As I mentioned before, we use the suspension adapt HEC293 cell line, the XP293F, as a cell culture platform. We have grown the cells in the BioBlue 1C single-use bioreactor controlled by our newest Siberia Twin bioreactor control system. So let me explain a little bit more the process in detail. So before running the experiment, we have to consider and prepare three important components: the inoculum, the bioreactor, and the controller. So let me explain a little bit more about the inoculum preparation. So this inoculum preparation will take almost around 2 weeks. We started with the cells expansion process in the shaker, as is shown in this picture. So we culture the cells in the New Brunswick S41i CO2 incubator shaker at 37 Celsius degrees, 8% of CO2, and 125 RPM of agitation. We obtained more than 900 million cells in the third passage. And during the expansion process, we kept all the inoculation density, percentage fill of the shape flask, and other parameters constant. So finally, we prepared inoculant containing 400 million cells in 200 milliliters of FP293F expression medium on each inoculation bottle. I want to highlight that here we inoculate, we run two experiments at the same time, and this is one of the coolest features that our newest controller has. But I will explain a little bit more about that later. So the second one is bioreactor. So the day before the inoculation, we have connected the pH sensor to Siberia to the bioreactor control system. We perform the calibration process according to the operation manual using buffer solutions of pH 7 and 4 as 0 and expand respectively. Then the pH sensor was disconnected and sterilized. After that, after being sterilized the sensors, we outfitted each BioBlue 1C equipped with a single pitch blood impeller with those pH sensors. And we inserted the pH sensors in the PG 13.5 port under aseptic conditions inside, of course, the biosafety cabinet. In addition, we also connect polarographic DO sensors, an exhaust condenser, a 4-gas mixing line connect to the gas sparge port, and also a silicon tubing extension connect to a different liquid addition port. So one that we have in the BioBlue, one for the inoculation, one for the base addition, and another for the addition of antifoam. Then we place the BioBlues 1C bioreactors in their respective temperature control block to keep the system at constant temperature. And finally, we add 800 milliliters of the medium into each bioreactor and condition for at least 24 hours under the parameters and set point list in the table that you can see here in the slide. Finally, 24 hours after conditioning, we inoculated both a BioBlue 1C single-use bioreactor with the inoculant, so 200 milliliters of this inoculant described previously, containing 400,000 cells per milliliter, with a cell viability more than 95%. We set also the temperature at 37 Celsius degree, and we have controlled the dissolved oxygen at 40%. Finally, the pH was controlled using the cascade that you can see here in this slide by basically using CO2 and sodium bicarbonate. Also, we took a sample from the bioreactor daily and measured the pH, the cell viability, and density, as well as the concentration of different metabolite offline. So this is our newest controller, Siberia Twin Bioreactor control system. It is capable of controlling up to 2 single-use or glass bioreactors, either individually or in parallel, using a wide range of bioreactor size with a working volume ranging from 1.3 to 40 liters, either way from microbial or cell culture. So the next step in the workflow is the plasmid transfection and the viral vector production. 3 days after the bioreactor inoculation, we reached the transfection cell density of approximately 3 million cells per milliliter. So we have decided to do the inoculation at 3 million cells per milliliter. Then plasmid, the two plasmids that we use at certain ratio were diluted in the medium, I mean the XB293F expression medium. And then in this case, we have used factory as a transfection reagent at certain radio. And then that solution, expression, the transfection mix was added into the plasmid, and the medium solution incubated for a certain period of time at room temperature, and finally added to the transfection mix. This transfection mix was added to each bioreactor. We have observed a rapid increase of cell growth in both bioreactors between days 1 and 4 of culture, as you can see in the right top picture, reaching a peak in viable cells density at 7 million cells per milliliter. I also want to point out that the process in both bioreactors show lower cell growth after transfection if we compare with the bioreactor process without any transfection. As I mentioned before, the information that you will find in the application note 447 of the last year, but essentially we were able to reach 13 million cells per milliliter in that case. Also, we have determined here the consumption of glucose and the production of lactate and ammonia daily during the whole run. Then finally, let me explain a little bit more about the isolation and the quantification. So in terms of the AAV2 capsid isolation, so 72 hours after the transfection, we collected 60 milliliters of the cell medium and the cells that were, of course, in the bioreactor, welding a bag with a cell line to one of our bioreactor liquid addition ports. So the cells were centrifuged, and we separated the cell pellet and the supernatant. And then we used AAV probe maxid purification kit. And again, I encourage you to find more information in the application note. And this kit was used to isolate and purify the AAV2 capsids in the cell pellet and in the supernatant, following the manufacturer protocol instruction. So following the viral particle isolation, we quantified the total amount of capsid in the cell pellet and in the supernatant using AAV2 Express ELISA kit. We have observed here lower AAV2 capsid titer from the supernatant compared to the cell pellet in the AAV2 production process from both bioreactors. We obtained a very similar AAV titer from both bioreactors, demonstrating an excellent reproducibility of the AAV production using our precision controller bioreactors. Finally, I would like to explain the second example that we have prepared for you today, and this will be the SARS-CoV-2 hexapro spike protein production. So the COVID-19 pandemic is ongoing and is far from it being over. And with the decrease of risk of exposure to new pathogen, more scalable vaccine productions strategy are needed to meet this global demand, as I mentioned before. So subunit vaccines only include part of the pathogen. For example, the COVID-19 vaccine contains spike protein, S protein, which is recognized by our immune system and triggered the immune response, including antibodies and white blood cells that will fight the virus after being vaccinated. So as you see, this is an schematic representation of the spike protein production and potential immunization. So starting from the production of the plasmid, after that using the plasmid for the cell transfection, the cells will produce the antibodies, and then of course this is an immunization, potential immunization cycle. The humans will get immunized, and then the whole mechanism is the same as I explained before. In the right side, you see a market analysis. That this market analysis has concluded that the global subunit vaccine market is anticipated to grow around 11% during the forecast period of 2022, 2028. In this regard, our company offers a wide range of product and technical guidance, such as these application notes, to help our customers achieve their goals. The entire workflow of the SARS-CoV spike protein production, which includes several steps, from the plasmid production, cell expansion, and plasmid transfection to a spike purification, titration, is represented here in this slide. The hexapro plasmid was produced in our lab. The plasmid host E. coli was expanded by fermentation using J flask to produce milligram level of plasmid for the bioreactor transfection use. We use the same cell culture platform, so we use the HP293F cell line, transfected the cells using the hexapro plasmid to produce the SARS-CoV spike protein. And finally, the protein was purified using strep-tactin resin and characterized by ELISA and SDS page, as you see. So let me explain a bit more in details the process. The inoculant preparation, bioreactor conditioning, and Siberia twin controller setup was identical to the AAV vector production example that I just shared with you. But in this case, we have carried out a medium optimization study and finally decided to use HEC, BIP, and X cell culture medium, as you see in the table. The other bioreactor condition remaining the same. So we inoculated both bioreactor with a cell density as before, so approximately 400,000 cell per milliliter, and carry out the transfection on day 2 at approximately 1 million cell per milliliter. As you see in this picture, we use we obtained 2 different grow profiles using, again, 2 different transfection reagents. In this case, we use Vectovir as before, and then also VectoPro that is specifically developed or designed for protein production. So when using Vectovir, a rapid increase of the cells growth was observed up to day 7 of culture, reaching a peak in viral cell density around 9.5 million cells per milliliter. In the case of VectoPro, the cell growth profile was affected compared to the Vectovir transfection. So reaching, in this case, 6.6 million cells per milliliter at day 8. So we would like to highlight here that both bioreactors show lower cell growth after transfection compared to the XP293F cell growth in 1 liter bioreactor under the same condition. As I explained before, that information that is explained in the application note 447 of the last year. So finally, we also analyze all the concentration of metabolites on daily basis. So every day until day 5 after transfection, 60 milliliters of sample containing the cells and the medium were collected using, as before, a bag, and this bag was welded to the bioreactor liquid addition ports. The cells were centrifuged again, and then the supernatant was collected. In this case, we have isolated the spike protein from the supernatant. Then this spike protein was purified by affinity chromatography. So after, you can find, this application though is not released yet, but then I can provide more information about all these steps later on. So after this purification step, the pure spike protein from the supernatant was titrated through the human SARS-CoV RBD ELISA kit. And this ELISA antibody pair detects SARS-CoV-2 regional binding domain, RBD, of the S1 subunit of the spike protein. Then the assay was conducted according to the manufacturing protocol instructions. The spike protein concentration when using FectoPro as a component of the transfection measure was higher, about 4 milligram per liter, compared to the bioreactor in which Fectovir was part of the transfection measure, in this case 3.5 milligram per liter. The spike is lower at growth post-transfection. And finally, we use SDS page to analyze the spike protein produced in both bioreactor. And as you can see here in the picture, both samples show high purity with apparent molecular weight of 190 kilodalton after compared with the standards. So I want to thank you. I hope you enjoyed the presentation, and I will hand over to David, and he will provide more information about our bioproduct solution.
Thank you, Jorge, for your presentation, and thank you all for listening so far. Before we will now start with the Q&A session, just a reminder, if you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and all questions that are not answered today, we will take the time, answer them via mail, and we'll get in contact with you afterwards. So to give you some time to ask the questions you might have for the different sections, I would like to show one last time the Savario Twin and the BioBlue bioreactors, single-use bioreactors Jorge used in his experiments. He just did show to you. So first, our Savario Twin, our latest bioreactor control system, which is a really nice system where you can control 2 bioreactors in parallel or also individually in quite broad size range from 1 liter range to 40 liter range. And our BioBlue single-use bioreactors you did also see during the presentation with a volume range from 100 milliliters to 40 milliliters. Not both. The BioBlue vessels are also available for fermentation processes, so in the BioBlue F versions with Russian impellers and other specifications compared to our cell culture version with the pitched blade impellers. So, and in the meanwhile, I did see that some questions showed up in the chat. As said, on the far left corner of the screen, there's the Q&A box. Type in your question, and I will start now with the first question that I can see here. And this is, what is the filling volume, Jorge, you used in the different experiments for the bioreactors?
Well, for a bioreactor, you should work about the minimum working volume of the vessel, so that you can cover the sensor. That would depend of the the bioreactor you're using, but basically you can work, as I said, above the minimum working volume, and then cover all the sensors.
Thank you. Another question that I can see in the chat is, why do you produce empty capsids if one of the requirements to produce a virus vector vaccine is to eliminate empty capsids?
Yeah, that's a really good questions, and I know that there is a safety issue regarding the percentage of empty capsids in the production. But our goal here in this application was wasn't to develop a vaccine candidate. So I want to say that the main goal is to help our customer creating simple solutions and workflow for vaccine applications in this particular. And we have developed an NAV production platform that can be easily customized to produce, in this case, prophylactic vaccines or therapeutic vaccines, or even can be useful for gene therapy.
Thank you. A question that just came in is, in your experiments, what do you think is the main limiting factor to scale up the process, so the AAV production, in a more robust and efficient manner? So the main limiting factor to scale up AAV production.
What do you Well, there are many factors that are involved in the manufacturing of AAV, and I think that having a good platform is important. In order to produce all these AAV capsids, and yeah, I think that this will be the main factor to consider, during the upstream process. Having the adequate cell culture platform or cell line platform or cell culture platform anyway to grow the cells and obtain the AAV in a good conditions. And I think that will be one of the most important factor to consider.
Okay. Another question is, why did you conduct a cell culture media optimization study on hexapro spike protein production?
Well, this is another interesting question, and there was also only a cost-effective related decision. So as you know, if you have to scale up the process, the cost will be important in order to to scale up the entire process. So that was only a cost-effective related decision that we took in the lab.
Okay, thank you. And last reminder for everyone to ask your questions. Otherwise, I will come to the last question I can see in the chat, which is, I am surprised to see the comparison between AAV collected from the cell pellet and the supernatant. Are AAVs not expected to diffuse into the cell culture medium?
Yeah, that's a really nice question. And unlike other AAV, so there are like many serotypes, AAV serotype, but in this case, I mean, we choose to produce AAV2, and the AAV2 capsids are really highly retained within the cells. And in this case, the cell digestion method, whatever you use, is needed to isolate the capsids. Yeah.
Okay, then there are no other questions in the chat. Thank you again, Jorge, for your time today. And before we go,
thank you.
Thank you. Before we go ahead, I would like to thank you all for joining us today and for the interesting questions you all put in the chat. Questions we did not answer, we don't have, doesn't matter. But if you want to rewatch the webinar, it's available on demand here on LabRoots. And if you then have questions, just type them in and we will still see them and answer them. Our next webinar on antibody production in Pichia pastoris, by the way, will be next week on Thursday, same time, same location. As well, if you want to register for our Eppendorf Bioprocess Spotlight newsletter to stay informed on upcoming events, visit our Eppendorf Bioprocess website, eppendorf.com/bioprocess, and get recent information on like webinars like today, but also information on events such as the upcoming Stem Cell Community Day in 2023. And with this, I say thank you and wish you all a nice and pleasant rest of the day.